Hello, everyone, and welcome to this live stream where we will be discussing the Resveratrol, Sirtuin, and NAD research and how they relate to our health. Now, the goal of this discussion is to have an open and honest and respectful look at the differing viewpoints and controversies around Resveratrol and Sirtuin. So, we're going to be moving through the timeline of research, starting from 1997 and working our way up to the present. And I do want to stress before we begin this discussion that before thinking about any supplements or interesting research, in terms of your health, it's diet, exercise, sleep, and meditation that always come first. So, joining me today, we've got Dr. Christin Glorioso, who's an MD and PhD and postdoc from MIT. She's worked in the Guarente lab, and that was the lab that was heavily involved in the 1990s and 2000s on the research around activating the SIR2 gene and how that might affect lifespan. It's also the lab where Dr. Matt Kaberlein and Dr. Brian Kennedy previously worked. We've also got Dr. Charles Brenner with us, who's an expert on NAD metabolism and its roles in health. And in 2004, he discovered the nicotinamide riboside pathway to NAD. So welcome, both of you.
Hi. Morning.
Hi, thanks for having us.
So yeah, the original excitement around resveratrol was somewhat to do with the idea that we could activate the enzyme SIR2 in yeast and SIRT1 in mammals, which was hopefully going to provide these lifespan benefits. So I think let's start by going through the research on sirtuins and if overactivating them is actually a good idea in the first place. So I believe, Charles, there's a 97 paper that you wanted to start this discussion off with.
Yeah, I think we could start with everybody in agreement that, in the 90s, in Dr. Guarente's lab, there was a lot of excitement and activity of using yeast. So Saccharomyces cerevisiae as a model for aging. And so this is a paper, Sinclair and Guarente. And in the previous couple of years, they had shown that extra copies of a gene called SIR2, SIR2, extend lifespan in this particular model. And let me just take a minute to explain this model, because how to use yeast to figure out how long cells or an organism lives, right? It turns out there's two ways. One way is you can grow the cells all to exhaustion, basically, right? So you grow them in a culture until they divide and use up all of the resources, and then you can keep that test tube or flask for days or weeks or months, and you can plate for viability, right? That's called chronological aging, chronological lifespan. And then there's the other kind that was being done in this paper, for example, by Sinclair and Guarente. And that's where you sort of install a graduate student or a postdoc in front of a dissecting microscope, and you look at how many times the mother cell can produce a bud, a daughter cell. And you remove those daughter cells, and then you score the number of times the mothers can divide. And in this paper, Sinclair, and that's the model that was being used. In this paper, Sinclair and Guarente acknowledged that those old mother cells basically don't exist. They're incredibly hard to find in a culture. Before there's a mother that has 20 bud scars, there's literally 2 million cells in the culture. And so the type of aging that they were looking at is the old mother, whether she doesn't really have a sex, but we call them she. Whether the old mother can divide a 21st time, but that's literally 1 out of 2 million cells. So there's no selective advantage for this type of aging of the old, mothers, because any cell in the culture can regenerate a whole nother culture. But people study yeast because they study yeast. I did yeast for more than 2 decades, and so I know this paper well. And essentially, what Sinclair and Guarente showed is that the old mothers can use SIR2 to divide a 21st, a 22nd, and 23rd time, because something called ribosomal DNA circles stays in the mother cell. And so they figured out that mechanism. But they knew that ribosomal DNA circles don't exist in other forms of life. They knew and acknowledged in this paper that there are very few of these old mothers, and that they didn't claim that it was a selective advantage. But this is a great paper from yeast. But what was extrapolated from this paper was that maybe SIR2 would somehow anticipate causes of death or aging in animals. And it generated kind of more excitement potentially than it was warranted. It was certainly reasonable to test the hypothesis that animals with more copies of SIR2 would live longer. And the Guarente and Sinclair labs and others tested that hypothesis. But given that there's no selective advantage for that old mother to divide a 21st time when 50% of the cells have never even been a mother one time, there's obvious limitations of this study.
So would I be correct in saying that it's great for yeast, but not exactly something that you can extrapolate to mammals, for
Well,
example?
it's actually even a little worse than that, because in 2004, I think, and I think you probably have the paper, Brad, Walter Longo, who's a very well-known and well-regarded um, aging scientist here in Los Angeles at USC, showed that deletion of SIR2, not extra copies, but deletion of SIR2 will extend lifespan in the other model of yeast aging, right? That chronological model of yeast aging. And this is selective, right? So if yeast has been around for 1 billion years, and yeast was here before there were any animals, and we're talking about conserved genes, so genes that are in yeast that are also in animals that might have a conserved function. Here, if you don't have SIR2, you have a SIR2 deletion, those cells, all of them in the culture, can live longer. And one would assume that all yeast are descendant of yeast strains that are resistant to desiccation and resistant to using up all of their nutrients. So here it's not 1 out of 2 million cells that has an advantage. It's every cell in the culture will have a longer chronological lifespan if it doesn't have SIR2. So that makes SIR2 not a gene that fights aging, that if you have SIR2 activity, that you're better ager. This indicates that if you don't have SIR2, you're a better ager in yeast. And this, as I said, is a selective trait, but this wasn't known until 2005, right? And in the meantime, there was so much excitement to test whether, extra copies of SIR2 extend lifespan, that people reported that it extends lifespan in worms and flies. And it wasn't until 2011 that we learned that individuals from 9 different institutions, led by Linda Partridge and David Gems, showed that you can't reproduce those results. That in flies and worms, that when you backcross those flies and worms, the extra copies of SIR2 don't provide a longevity advantage. So that's really where the whole SIR2 story got off the rails.
So thanks, Charles. I think I'll turn it over to you, Kristen, about that original sort of SIR2 research. So is there anything that you'd like to add or comment on from what Charles has said, and any other papers that you wanted to go through yourself?
Yeah, so I just want to say I study humans, so this is far from my area of expertise, yeast. But and also was in the Grenty lab many years after this paper was published. But in thinking about this, I mean, I think this is kind of an interesting dichotomy in science, right? Something that's extending lifespan, and replicative yeast, and possibly not extending, going in the opposite direction in chronological lifespan in yeast, and how do scientists go in and figure out who's right or what the nuance is, or how do you resolve that discrepancy, and how do you move forward? And I think, just for the audience, to put this in a big picture context, right? The real reason we're doing all of this, or I mean, there's multiple reasons people do things. But one reason that I think is what the audience is probably interested in is how do you go from there to drugs that are going to help people live a healthy life, right?
Agree.
What is the role of resveratrol, right? So the reason people start in yeast and start in lower organisms is because they're easy to screen. So right, Sirtuin was found by a screen for Lifespan originally because how do you do something high throughput to find even a starting point, right? So that's where we started. And then there was this, all right, well, if it works in yeast, or there's some controversy in yeast that's still being resolved, obviously, and maybe, Charles, Dr. Brenner can talk a little bit about where that controversy is now and what's happened in yeast since then with Sirtuin. But yeah, there was then people who tried this in flies and in worms, and there was a 2000. So Grenti Lab showed that there was extension with overexpression of Sirtuin in worms, and then there was this 2011 paper, right, where Partridge Group came out and said there was an extension in flies and worms, and then Grenti Lab came back out and said, okay, we did this 20 times. There was a little bit of a background error in that first paper, but here we've done an extra good job showing that it does extend lifespan to a lesser degree than the original paper said, but still extends it. So there's this back and forth, right? As we go up and as we go up the phylogenetic tree, and as we try to work our way towards finding drugs that are going to help people.
So a lot of this, sort of comes back to the premise, right? And Christin, I think we agreed yesterday we're all going to call each other first names. So 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 Christin said, there's a discrepancy in yeast. I would say not really discrepancies. So it's two different models, right? So chronological aging, where it's all of the cells grown to stationary phase and how long do they survive, right? So then Sirtuin is bad. So if you have Sirtuin, you have a short lifespan and cannot be extended by calorie restriction. In the replicative model, where it's this 1 out of 2 million cells for which there's not really a selective advantage, Sirtuin is good. Those two things can both be true, right?
Mm-hmm. Yeah.
But the premise, this is important. The premise was that there might be genes that are conserved between yeast cells and animals that control the process of lifespan extension with, in response to calorie restriction. And that premise has basically been defeated because when you say a conserved gene, it means that a gene that has to stay doing that function in other organisms. So for example, citrate synthase in yeast is there to catalyze a reaction between oxaloacetate and acetyl-CoA to make citrate, right? It's same in E. coli, same in yeast, same in humans, right? And so the idea is Sirtuin is a longevity gene, and it's conserved, meaning that organisms would have dropped off of the face of the earth if they didn't have this gene, and that's why it's so fundamental. That's why it was in the yeast gene set, and that's why it's in the worm and fly gene set and the mouse and the human gene set. That's what a conserved gene is, right? But Longo's experiment in 2005 shows that it's not conserved as a longevity gene. If yeast still has Sirtuin genes, it must be because of some other property of the Sirtuin gene. Actually, Sirtuin is also required for haploid cells to mate with each other. But if it was all about survival, right? If it was all about survival, yeast would have lost the Sirtuin gene. And by the way, that mechanism that was described so beautifully by Sinclair and Guarenti of repressing ribosomal DNA circles is not a conserved process. So the premise that it was a conserved process and that it's a gene there that is in other forms of life to mediate the effects of calorie restriction has been debunked dozens of ways, right? And I think that we're both interested in human now, right? You and I, as well as Brad, who's a physician, and it doesn't seem like sirtuins add a lot of value in terms of human medicine. There was a company formed initially to test whether SRT 501, which is a formulation of resveratrol, could activate SIRT1, and maybe that's going to be a drug. And folks were in the New York Times claiming that it's going to treat diabetes, and as a side effect, people were going to live longer, right? And that and other compounds were clinically tested. There were some safety problems. They don't extend lifespan, you know, in model organisms. They don't look like promising drugs. And I think that we've kind of, tested the premise, and the premise and the hypotheses have been falsified. And, now it's claimed that sirtuins mediate the effects of NAD. And as a professional NAD researcher, I can tell you that they don't. And so, that's, I think, where the story stands, and that it's a nice story, but there's not any scientific validity to the idea that sirtuins are longevity genes.
So I mean, I think there's kind of a lot there, and we jumped ahead a little bit, but I think um, are sirtuins longevity genes? If that's the question, I mean, I think part of what you're saying is that they don't have just one function that's mediating caloric restriction. Well, lots of genes don't have just one function, right? I don't think that is makes them not doing the other function. So an example of this, right, is cell cycle. So cell cycle genes were found to, you know, be involved with cells dividing, right, in peripheral tissues and in tissue culture. And but then what is the cell cycle genes doing in non-dividing neurons? They have a different function, right? So I think the sirtuins, by the way, right, there's one sirtuin in yeast, but that turns into 7 sirtuins in humans, right? So they divide and have a whole family of sirtuins. Each one of them has a different place in the cell, and each one has a different function. And so it gets quite complicated in terms of what they do. So uh, are they extending lifespan in higher organisms? I think, in male mice, it was shown that SIRT6 is possibly mediating lifespan. There's been a lot of papers showing Healthspan benefits to overexpressing sirtuins and in some cases knocking them out. So, right. So,
Right.
and that's not just true of sirtuins, right? That's true of a lot of these so-called longevity genes. I don't know that I would say that to be called a longevity gene, it needs to have only one purpose, and that purpose is
No, I agree. I agree. It doesn't have to have one purpose. I said that the premise that they were conserved to extend lifespan doesn't hold up. That, but and we've really known that since 2005, because where there's a selection for chronological aging, and there's not a selection for 1 out of 2 million cells to divide a 21st time, right? We can agree on that, right? That literally the oldest cell out of 2 million, whether that divides one more time or not, the yeast culture doesn't care about that. But in chronological lifespan, um, it's the survival of the cells over time, right? That's clearly a selective trait. So by 2005, we know that even in yeast, SIR2 can't be conserved as a longevity gene because deletion of it has a longer lifespan, right? And and then we know that like SIRT1 is essential in some mouse strains, is not essential in other mouse strains, basically as a bone growth phenotype, right? and but the cell can, I mean, the mice, you can make the mouse, right? And it lives a normal lifespan, and then you overexpress SIRT1, and they did not get extended lifespan. Now, the thing that is important, though, is that the amount of attention and expectation that arose out of this amazing story, because um, the principals are excellent scientists and amazing storytellers, right? And so the story was that we have found the central mediators of lifespan and the central mediators of caloric restriction, which I think that everyone involved in this debate, if it's a debate or discussion, I think we all agree that these are not the central mediators of caloric restriction and lifespan. Would you agree with that, Kristen?
I mean, what does that even mean? What process is the central mediator of caloric?
Yeah. Right. But you wouldn't, if someone said, is there a single mediator of lifespan or caloric restriction? We might not even like the premise of the question, but we certainly wouldn't nominate any gene in the sirtuin family.
Where was that said, though? Is that?
It was said hundreds of times. It was said, in promoting Sirtuin. It was said in hundreds of NIH abstracts. It was said in hundreds or thousands of review articles, conclusions of papers, that these are the central players in lifespan, and that there's an activator, namely resveratrol, of Sirtuin, that is going to allow people to live to 150. It was said thousands of times. It's well recorded. It's in a best-selling book today.
Right.
Right.
So, well, I don't know. I don't have the book memorized. Actually, I actually, no, sadly, have not read that book. I probably should. But I think this is a question of language, and this is one that I think we're trying on a high level to deal with.
Yeah.
In general, in science communication.
Right.
I think scientists, maybe most scientists, right, are very careful about the words they choose and the way they describe things to try to be precise, to try to not overstate conclusions, to try to be very accurate. It's a matter of scientific integrity.
Right.
Then you get into the world of what's printed in a newspaper headline, right?
Yeah.
And it's very different. Most scientists are upset about that,
Yeah.
And
yeah, but not all.
So cancer, right?
So for example, in 2007, so our lab discovered the NR kinase pathway, as Brad said, in 2004, and we knew from the work in the Grunty lab that SIR2 is an important mediator of this replicative lifespan in yeast, right? So we put NR into that assay. And the reason why I know it's the old mother cell is that, it was done in my lab by Peter Belenke. And we reported in 2007 that despite high glucose, so not calorie restricted conditions, we could double yeast lifespan with NR. And that it depended upon the NR kinase gene and the other genes that take NR to NAD. So we showed that we could extend yeast lifespan with NR in 2007. But I never got out ahead of my skis and said that because, SIR2 and its homologs are the central mediators of lifespan in humans, that NR is going to be a human longevity drug. And I won't say that today, because human aging is different than yeast aging. Human aging is different than mouse aging. Human aging is different than macaque aging. In primates, males and females are fertile throughout their lifespan. In humans, there's a really, really interesting exception to the reproductive capacity going along with lifespan with women, right? So women have menopause, usually in their fifth decade.
And men to a certain extent, I will say that.
Well, no, men have a declining sperm production capacity throughout their life, but it doesn't fall off a cliff, right? So human women are an exception to a very broad rule that says that animals live as long as they can reproduce, right? And in 2007, nor do I think today, that any gene in yeast can anticipate the causes of aging in an animal and especially not in a human, right? Because even primate gene sets don't anticipate the delta between female reproductive capacity and female longevity in a human being. There's some human specific genes that confer that involves, the roles of grandmothers in the 300,000 years of human evolution. So biology is complicated, and storytellers, and it's not only, newspaper headline writers that take things and hype them up. It's some individuals in science that hype things up and say that because we found it in yeast, we're going to find it in worms, and then publish things prematurely that turn out to be not reproducible. And then there's other people that don't even ever retract or say things or admit that things aren't true. So resveratrol doesn't actually, activate SIRT1. It doesn't activate yeast SIR2. It doesn't extend yeast lifespan. Um, Matt Kaberlein showed that it doesn't activate SIRT1. It doesn't extend yeast lifespan. And there are folks today that are saying, "Oh no, resveratrol is a SIRT1 activator," and there's a huge global market for resveratrol for exactly that reason. And there are people that are still saying that SIRT genes are conserved as longevity genes, despite the fact that even in yeast, that chronological lifespan is longer without it. So it's not only headline writers that do hype. It's folks in the scientific community, and I would argue that hype hurts.
Right. So okay. So that's really like, I think this is a version of always punished, right? But there's this two different ways of communicating. There's the way that scientists communicate with each other, right? And then there's the way that people that are not in science communicate about the science. And so there's pros and cons of both, right? I'm an academic. I'm currently, right, trying to learn how to communicate in a way that is going to resonate and not bore the pants out of, people that are non-scientists, and have them actually be excited about what we're doing, be excited about the future of science. We have decreasing science funding in this country. We're increasingly siloed. So how do you communicate both effectively and with passion, and to show the excitement behind science, while at the same time not saying things that are overblown or inaccurate, right?
100% agree.
And
Except that science funding is increasing federally as well as privately now.
well, not as a percentage of R&D. I can show you those figures, but anyway, it's so some of the best speakers, right, that I've seen. So if you go to the big Society for Neuroscience and watch the keynotes, which I sometimes do, and they're amazing, right? And they're some of the most famous scientists in the world.
Yeah.
And they are able to take a field that's not my field, right, and tell me a story that is both scientifically accurate and simple enough for me to follow, being not in my field, and inspiring, right?
That's what we all aspire to.
Right. So that's what we hope for. And I completely hear you that like some of those claims may have been too simplistic, right? They're made for investors, they're made for the general public, possibly even, a lot of times people that are writing stories, they don't want a complicated science headline, such and such may have a role in, a specific type of yeast lifespan extension and this part of caloric restriction. Like, that's not a headline, right? So it's like, how do you make a headline that is something that people are going to understand and be excited about?
Do you want to know something really, really funny? There's something called Betteridge's Law. And Betteridge's Law states that any headline that is stated as a question has an answer, and the answer is no. So, for example, if the headline is, "Could Sirtuin genes be the key to human lifespan?" And there have been innumerable headlines such as that. The answer to that question is no. Could this phytochemical eliminate fatty liver? The answer is no. Could
Has
we
somebody
all
studied
live
that?
forever? The answer is no.
Has
That's
somebody
better.
studied that and looked for outcomes on that stuff? I don't know. Is anyone proven betterage is betterage?
No. It's just it's an aphorism, right?
Mhm.
But it's generally true that when the headline writer knows that they have to state it as a question rather than saying, um, Harvard University, City of Hope, UCSF scientists have shown that a phytochemical is the key to fighting fatty liver, right? They won't write it as a declarative sentence, so they write it as a question, and 99% of the time the answer is no.
Mm. Yeah. So I think to bring it back to what we're interested in, which is how do you move forward with human health span extending drugs or interventions, right? I whether or not sirtuins are activated right by resveratrol, right?
They're not.
Whether so there's been a big back and forth in the literature. I don't know. I read a bunch of review papers. There's this, there was stuff about a fluorophore and then amino acids, and then
Do you want me to cover that as a biochemist? You want to go through that because that's sort of what Brad promised out of this hour. We could do that.
I think that would be a good segue into that. I think there is still some debate
Okay.
from what I'm gathering from you two about sirtuins and the merits of whether we should be overactivating them or not. I think, yeah, Charles,
Should we do it?
am I correct in saying that there's probably no benefit of overactivating the SIR2 or sirtuin genes in mammals? And Christin saying, well, that the literature is still open. Is that where we're at with this stage of the discussion?
It sounds like we have an agreement. Let's see if I can summarize. We have an agreement that scientists should not overhype their studies, right? We have agreement that
I think they should hype them appropriately, but find a way to say it in a way that's not going to bore people and decrease science funding.
Ooh, I have a problem with hype. There's a way to
hype, right? It doesn't have
accurate
to
hype.
be.
Well, excitement. To conveying the excitement is good, right? But to me, hype means to amplify something in a non-evidence based way. So that's why I can't get behind things that are hyped.
Okay, yeah. I mean, I don't think it should be in a non-evidence based way, but it should convey the excitement. And also, all of these journals are behind paywalls. How are people supposed to know what we're doing at all, right? And then you read them, and even experts in the field can't follow them sometimes.
Yeah. Okay. So we agree. Is it okay if I say hype hurts then? If we say hype being defined as non-evidence based statements that are likely to get people to think that a problem has been solved or that
Yes.
so we agree on that. We agree that even in yeast, cells live longer if they don't have SIR2. So it's kind of a head scratcher why you would expect animals to live longer if they have more copies of SIR2. But we haven't talked about resveratrol yet. So Caberline actually, and he had some impressive co-authors on these papers, Stan Fields and Brian Kennedy. They were interested in yeast, right? And they were interested in the SIR2 gene. And Kennedy and Caberline were both in the Grenti lab, and they both worked on
Sinclair, by the way.
and Dave Sinclair, of course, and so they wanted to see whether resveratrol extended lifespan in yeast, and they reported it doesn't. So they're using the same assay that David did of replicative lifespan, this old mother thing.
Mhm.
And so that's in this paper, JBC paper. And then further, um, and now I don't want to get too geeky as a biochemist, but you see in this abstract it says fleur de lis. Yeah, lys is a amino acid, lysine. So there's a particular peptide that was synthesized by Conrad Howitz. It was in David Sinclair's first paper on resveratrol as a sirtuin activator. And basically, this paper, Caberline, showed that fluorescent dye is the thing that SIR2 is interacting with. That human SIR2 and yeast SIR2 interact, and that the resveratrol effect is something that occurs in a test tube, and it doesn't actually involve an, a physical interaction with a sirtuin gene or sirtuin gene product. Then years later, David came back, Sinclair came back and said, no, I've mapped the amino acids in human SIRT1 that you need these amino acids in human SIRT1 to interact with resveratrol. And the resveratrol thing is real, because if I mutate these amino acids, I don't get the activation of resveratrol, right?
Right.
So that's Hubbard. Thank you. Brad is like right there with all of these papers. So, so according to Hubbard, it's these particular amino acids that are specific to human. And the problem is, if you go look for those amino acids that Hubbard and Sinclair found in human SIRT1, they're not in yeast SIRT2. So then something's got to be wrong. David could not have found resveratrol the way he describes resveratrol according to his current explanation of how resveratrol works. So it's constant goalpost shifting. It's first saying that resveratrol extends lifespan to yeast. That can't be reproduced. Then resveratrol activates the SIRT1 enzyme. That can't be reproduced. Then he says, well, no, it does activate human SIRT1, and here's how. But now he's moved the goalpost around that it couldn't have been where it initially was. So multiple bodies of this research really should have been retracted. And I think that would be a much more straightforward and honest way of dealing with the facts as we now understand them.
Well, I think somehow this has turned into become like a moratorium on David, right? And I think like it's, there has I don't know what to say, because I wasn't there at the time when all of this was happening, but there's a back and forth of papers, where there'll be a paper published by David Sinclair, then there'll be a response from Caberlin, and this back and forth and back and forth. Where are we with where resveratrol is in terms of activating the sirtuin genes? I'm not sure that's even resolved yet. From what I was reading last night, just in the review papers that Brad sent around, there's this question. Well, maybe you need multiple copies of resveratrol. Maybe in vitro, you need the N terminus is not there, and that's why people aren't seeing activation. And so I don't know where the state of this is, but where it was left did not look clear to me.
In my view, there's sort of like three invalidated premises. One is that you'd want to activate SIRT1. Two is that resveratrol is an activator of SIRT1. And three, that resveratrol would be available to a human being when taken orally. And all three of those things are wrong.
I don't know that I don't know, Brad, in that review paper, it looked like people were saying it was bioavailable.
It's not bioavailable. It's bioavailable to a mouse. A very, very high dose resveratrol has effects on overfed mice. We know that. That's been reproduced in other laboratories. By the time that Johan Auwerks, who I know you sent me some papers from Johan, was working on that, the last papers from Johan showed that it doesn't depend on SIRT1. It goes through the AMPK pathway because resveratrol is a stressor and doesn't depend on SIRT1. And so ours is basically consistent with all of the other stuff, saying that resveratrol has a totally indirect mechanism of action that doesn't depend on Sirtuin. And 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 again, the activities were in overfed mice. And
Well, there's a
yet,
lot of overfed people. I just want to point out.
yes, but resveratrol bioavailability is nil in people, um, which is why Sirtuin, made a microcrystalline formulation of resveratrol to increase its oral bioavailability, and which is why they looked for novel compositions of matter that would potentially activate Sirtuin. And the Pfizer group in 2009 showed that neither resveratrol nor the other proposed Sirtuin activating compounds from Sirtuin are direct activators of Sirtuin, that when you knock out the Sirtuin gene in mammalian tissue culture, you still get the effects of these compounds. They don't depend upon Sirtuin. They don't bind directly to Sirtuin. And so the whole thing has been falsified.
I mean, I've been hearing this story for a long time. I mean, there's a point of view that wants to paint David and his research as all wrong, but I don't think the literature bears a more complicated picture, in which there's this back and forth. Other people are publishing on aspects of it. The Brenner lab, the Aurix lab, Shinamai's group showing, positive results in specific subclasses of neurons, and there's a whole body of research on this. And I think is that it's become this personal battle, between scientists, but what should be conveyed to the public. I think it's a disservice to say that resveratrol has no good effects, whether or not it works through, sirtuins or not. I'm not sure that even really matters. It works through M kinase. It works through all kinds of other pathways.
It does matter because premise matters, right? So, for example, again, if the idea is that Sirtuin is a longevity gene, is what is stated over and over again, and therefore activating Sirtuin will promote longevity pathways. That the headline story, and it's in, like I say, it's in a book, says that we discovered longevity genes that are conserved all the way from yeast to humans, and we have activators of these longevity genes, and they work in humans. And if you look on page 304 of my book, you can see what my practices are, which involve a gram of resveratrol in the morning, with yogurt, in order to activate my Sirtuin gene. And if you go back, further, you see claims that we're going to live to over 150 by doing this. And so, if it turns out that that Sirtuin is not a longevity gene, then why would you be looking for activators of Sirtuin? And if it turns out that resveratrol is not orally bioavailable, or even worse, that it causes substantial toxicity, such that GSK, which paid a lot of money for this technology and tested it clinically, had to suspend clinical testing because of adverse events and no signal in any of the things that they were looking for, you'd have to say that this is a falsified hypothesis.
Well, I don't know. So there were a compound, right, made by a company called Sirtuin, which was a company started by David Sinclair, that was meant to make activators, right, of the sirtuins in order to treat healthspan, basically age-related diseases, right? And those compounds were patentable, and they went into a clinical trial. Now, did GSK buy Sirtuin before or after the multiple myeloma trial? That I don't know. Right? There was a multiple myeloma trial where there was toxicity.
Yeah, I thought it was. I thought they bought it before.
Trial. Yeah, they conducted that.
They conducted the trial, so they bought it. So So there was a small trial, right, of less than 50 people. I don't know how many people that had some side effects, right, including there's diarrhea and vomiting, and that led to renal failure in people that were already pretty sick, right? They had cancer. So, and they stopped the trial. Does that I mean, that's terrible, right? But does that equal sirtuins aren't
It was
longevity?
largely
A leap.
the thing is that they had many therapeutic, you know, areas at GSK that they thought that these sirtuin activating compounds were going to address, right? So prediabetes, diabetes, cardiovascular health, ocular health, and other things. And basically, none of it panned out. And mice with extra copies of Cert1 don't have supervision, um, super hyphen vision. They don't live longer. They don't have improved cardiovascular health. When Dr. Grunty, knocked out the Cert1 gene, he said the thing is
Well,
very
he did.
highly expressed in the pancreas, and that what happens in the pancreas of the SIRT1 knockout is overexpressed UCP2 with an uncoupling protein. So how is it that when you know that loss of SIRT1 gives you higher mitochondrial activity because you have higher expression of uncoupling protein, how is it that people can still go around saying that there's going to be higher mitochondrial activity if you activate SIRT1 when it's actually deletion of SIRT1 that gives you higher mitochondrial activity due to
Well,
experience? So there's just the problem is it's all story. You know, there's a saying, sort of all hat and no cattle. And I think that's what the Sirtuin story is right now. I think that
well, I think I think they're separate issues, right? All of these are separate issues that we're weaving into like a narrative here. But I think whether or not resveratrol works or has benefit is a separate issue from UCP5 and mitochondrial function. I mean, that could be cell type specific. We could go on forever why sometimes that's up and sometimes that's down, right? So I don't know that these are a linear story. I think, if we hang it in whether or not people should take resveratrol, that's the bottom line, right? I personally don't take resveratrol. I eat blueberries and grapes and stuff and try to increase my consumption of foods that I think probably could have some benefit, right? But should that be a line of research that's pursued? I think that's the bottom line. And I think one trial with one drug that had bad side effects doesn't definitively answer that question.
yeah, we could get some people from GSK that would potentially talk about the amount of research that was done and the degree of disappointment in sirtuins as activation targets. Um,
Well,
you know,
I wish that was published so that we could see that, right? Because we
well,
don't.
so that okay. So let's talk about that. So let's talk about confirmation bias and model bias, right? Because you're interested in science communications, and so what happens when someone publishes an exciting result? What happens when someone publishes an overhyped result, right? So when it was claimed that sirtuins are conserved as longevity genes, which I think we've established that they're not, even conserved in yeast as longevity genes because deletion of that gene gives a longer lived yeast, and that's a selected trait. Um, but when you claim that they're conserved as longevity genes and you make the story around it, then you shine this huge light of, okay, this gene, or like you said, 7 sirtuin genes, these are they were promised as the central mediators of calorie restriction, benefits of calorie restriction. They were promised as the central mediators of longevity, right? So that means resources went to those genes, right? And those targets that could have been, looked at all other things, right? You
I'm
said
sure
that.
a lot of researchers went to tour as well, right? And other gene longevity.
Sure. But but when something becomes hot, you know, scientists use the term sexy. I don't know whether it's really sexy, but um, it becomes, it's hot, it's considered exciting, it's hyped, and then people start looking at it, right? And then, like for example, the SIRT1 gene was simply overexpressed in a mouse, didn't extend lifespan, and then somebody said, "Oh well, maybe you have to just express it in one cell type." And then like dozens of different groups expressed it in different cell types, and it didn't extend lifespan in like 9 of those cell types, and those results don't even get published. And then somebody finds, oh, there's one particular neuron that if you have extra copies of SIRT1 in that neuron, then it extends lifespan. So that's confirmation bias, means because it's an exciting result, you can publish that positive result, but most of the negative stuff doesn't even get published. Like you said, we don't know exactly what GSK did. We know that they spent billions of dollars after they, invested 720 million in this program, but we don't have a lot of output from that, right? Because pretty much everything was negative, to the point that and for full disclosure, because I developed the nicotinamide riboside technology when I was at Dartmouth, I was on the scientific advisory board of Sirtuin, so I saw this stuff pretty close. I mean, I was an expert biochemist that was available to evaluate whether or not they could find compounds that activated, SIRT1 without the presence of the fluorogenic reporter group. John Deneau was on the board. Um, um,
And you guys
other,
also, I mean, being on the board also means there's equity, right? So you guys lost money when Searchers tanked, right?
I Sirtuin was considered to have succeeded because Sirtuin was able to sell itself to GSK.
Required.
So Sirtuin, as far as Sirtuin was concerned, as a biotech startup, they feel like they succeeded. Now, when pharma took that on, right, and pharma made $720 million investment and then invested billions of dollars after that, GSK feels like they got burned because frankly, they didn't really do their due diligence, and they should have paid more attention to critics and the substantive problems of whether or not the compounds hit their targets. And by the way, when I was an advisor of Sirtuin, and like I say, very capable of determining and helping their scientists determine whether they have direct activators, they were actually having meetings where they were keeping advisors like myself from seeing the actual data. Okay, so the play of Sirtuin largely seemed like, let's create a hyped story, and that's why I say hype hurts, and let's promote the heck out of this thing. It was constantly in New York Times, Newsweek, Time Magazine, Washington Post, and other places as a startup company that was going to develop longevity drugs.
Right.
And then GSK is a big traditional pharma that made this radical, play into, years before Altos, a radical play into the longevity medicine, if you will, right? And they feel like they got burned because the things just broke down, like the non-reproducibility of the even the worm and the fly life extension results, the non-reproducibility of resveratrol as a direct activator, the non-SIRT1 dependence of the SRT 1720, which is a Sirtuin composition of matter, that the Pfizer group showed that those don't depend.
I mean, first of all, I can't comment on what happened at GSK, right? You have some inside knowledge of that
Yeah.
I can't deny or verify, because that this is part of what happens at pharmaceutical companies, right? We don't know what's going on because it is behind um, NDAs and everything else, and people don't talk about what's happening. So, and it's not published. So I can't really confirm or deny any of that. But I can say from a high level, how many drugs work, right?
Very few.
How many drugs work, right? There are, you go in to these companies with the knowledge that there is a low success rate, right? Sometimes things don't work out. Is that necessarily the fault of the investigators? I don't know. I mean, does that mean that this entire class of genes or class of drugs is a total red herring? I wouldn't go that far. I mean, I think there's promise still in this stuff, from what I can see, from what other groups are publishing, from what's out there in the literature. Should we throw out bottom up approaches entirely for screening for drugs? I personally take a top down approach. I start in human for that reason, but I think there's a lot of scientists out there that start in worms or yeast or screening for drugs, and sometimes they don't make it all the way up the phylogenetic tree, right?
Right. Agreed.
But I don't think that means there have been Nobel Prize given out for discovering genes for diseases, right? That
Sure.
that have been discovered in C. elegans, that have been discovered in other organisms. So
Right.
I don't think we throw out that approach entirely.
No, I wouldn't throw out that approach. So,
I just wonder if I can just interject here. I just wonder if we can see if we can get a little bit of agreement. So, coming back to the idea of trying to overexpress or overactivate sirtuins. So, I think we can agree that there's certainly debate within the literature, and from what Christin you've been saying, if you read and for the viewers, if you read different review articles that are published and peer reviewed, some of them will say yes, resveratrol does activate Sirtuin 1, and that by activating Sirtuin 1, you can get these lifespan extension effects. Whereas it seems that if you go through the data yourself, and that, Charles, that's what I think you've been doing, trying to explain that story, is that, if you look at it, it does seem that the hypothesis that if you overactivate Sirtuin 1, you probably won't be seeing these lifespan extension effects. Is that something that we can agree on, and then we can hopefully move more towards resveratrol, and whether possibly the other targets of resveratrol might be worth further investigation?
I'm not even completely sold on that, because the last I was reading last night was 2015 papers, and it was saying that you need 3 different molecules of resveratrol plus the N-terminus, and then it activates sirtuins. So that's the last I heard. I don't know what the state of that is now, but
So what about coming to the idea though of activating Sirtuin 1? So where at the moment, Christin, after this discussion, where do you sit with trying to activate Sirtuin 1 in terms of how that relates to human health?
activating sirtuin 1 in human health.
Yeah.
So do you think
Brad
that
is
would be
saying if you had a better compound that was for sure orally bioavailable and
Didn't have side effects, and was it pure
no side
acid
effects and would activate SIRT1, do you still see the value of that as a potential human medicine?
I think that's unlikely to work.
Yeah.
I mean, the as was mentioned, overexpression of SIRT1 has not extended mouse lifespan. Mice aren't people, right? There is some work with other sirtuins that doesn't. SIRT5, SIRT6, maybe activators of those would be helpful. There's a lot more that needs to be done to understand the roles of all of these things, right?
Okay.
But
Sure. So overall, can we agree that activating or overexpressing Sirtuin 1 in humans is unlikely to give lifespan or health span benefits? Would that be a fair thing to say, Christin?
so I feel kind of like that, questionable about that. I mean, that would not be approach I would take. I think that's lower down on the things that are likely to work. But for me to say that definitively, I'd have to see a Sirtuin activator in primates showing no benefit. Because how else are we going to answer that question? Mice aren't people, right? There's back and forth on cell type specificity
But
and
if
what.
it's already failed in a mouse, would you spend, hundreds of thousands of dollars, millions of dollars to test it in primates?
I wouldn't start there, right? But here's the thing: mice are not people, right? So human brain aging and mouse brain aging if you look at the transcriptome changes between the two with age, there's a correlation of less than zero point 0.2.
Mhm.
So humans aren't mice. So I
Right. So to me, it sounds like, and by the way, I think that we could probably also agree that aging is very polygenic, right? There's
Yes.
thousands of genes, right? So there's thousands of genes involved in the function of our brains and our liver and our musculoskeletal system, our respiratory system, our circulatory system, right? And so the idea that activating or inhibiting a single gene would promote human health span or longevity is
Mm.
pretty much a long shot, right?
Well, I do think that there are probably upstream genes, right? Probably more than one upstream gene. There's probably, I don't know. So we're working on this in human brain, right? And so we found regulators that are upstream of human brain transcriptome changes with age. And so we do think that there are genes that are functioning as master regulators.
Hmm.
Not one gene, right?
Hmm.
But I think that's possible. I do. I think you have to affect the whole program or
Hmm.
the part of it
You'd
that
have to affect the whole program, yeah.
yeah.
And I think that would, I think, be a nice segue into resveratrol, because I think many people would agree that resveratrol is a so-called dirty molecule, and that it affects a lot of different things. And I'm not
I
sure if
don't
we
think
can
that
get,
makes it dirty. But go on.
well,
Yeah.
it's often referred to as a dirty molecule because it's not, it doesn't just have one clear target. That it does affect a lot of different things within the cell and within the body. So I I was hoping that we could get a little bit of agreement around the merits or lack thereof of Sirtuin One, but um, I think it would be good to move the discussion forward to more what the potential benefits or lack thereof of resveratrol supplementation might be, because there is certainly some debate, and I probably sit in the camp that resveratrol isn't a direct activator of Sirtuin One, but given that we can't agree whether activating Sirtuin One is a good idea or not, I think let's move the discussion forward to possibly the other targets that resveratrol may have. And I think, Charles, you mentioned that it might activate AMPK, similar to what metformin does. So that's possibly where we can pick up the conversation.
But the problem is that it's not orally available in humans. So to me, it's not much of a useful conversation. I'm really surprised that resveratrol advocates are still talking about it. It to me, it's been, as I said, disproven in terms of all of the premises that made it interesting. It's not humanly available. It doesn't activate SIRT1, and then to me, there's no evidence that activation of SIRT1 is something that you'd want to do. So I don't even fully understand why people are interested in it.
Kristen, what do you think?
Well, I think resveratrol does have. I mean, it targets AMPK and a bunch of other things. I don't know if you can pull up that review. There was one of the review papers you had on resveratrol towards the end. It had a paragraph that said that that it is bioavailable. So you know, I don't know. I
Well,
don't know.
pterostilbene was developed because it's more human bioavailable than resveratrol, but there are published data saying that pterostilbene gives a time and dose dependent increase in low density lipoprotein cholesterol, which is bad for human health. So, and contrary to the idea that pterostilbene is a SIRT1 activator. So, I don't understand why people,
Yeah, pterostilbene I don't think it was not made. It's naturally occurring
yep,
in blueberry skins. No one made it.
right, right. But it's sold as a supplement. And the company that I consult for, Chromadex, I'm the chief scientific advisor, had a proprietary formulation of pterostilbene. And when we looked at human literature that was coming out and saw that pterostilbene was increasing low density lipoprotein cholesterol, we shut down our business because we thought that this is a potentially harmful compound. There's no reason that people should be taking this.
Yeah. Okay. I mean, I'm just looking at the wiki page on pterostilbene. So it's found in almonds, berries, blueberries, grapes, and vines. A randomized double-blind placebo-controlled trial of healthy human subjects given pterostilbene for 6 to 8 weeks showed pterostilbene is safe for use at dosages up to 250 milligrams per day. It's a chemical to resveratrol, FDA status in 2007, approved synthetic.
There are two papers showing that it increases low density lipoprotein cholesterol in people. Like, they didn't have adverse events in 6 to 8 weeks, whatever it was, but we know the association between low density lipoprotein cholesterol and atherosclerosis and future cardiovascular events. So it's not healthy to take.
Yeah, I don't know. I mean, I think it's we can go in. I don't know how to comment on that because we can go into the weeds of each of these papers. It could be a battle like forever, right? In terms of what you want to cite. I think this is the problem, is that there's a body of literature for and against each of these things. If you read the reviews, they're not coming out with a consensus, right? And so if you only talk about one side of the literature and ignore all the other side of the literature, then you can support a conclusion, but that's cherry picking. So, I don't feel comfortable coming down on one side of this or another at this point. I mean, there's things that are clear to me in the literature, and there's things that are unclear. And this story, to me, is still unclear.
I suppose that's the thing though, Kristen. When I see patients in the clinic, so say if I'm seeing a newly diagnosed type 2 diabetic, the first line of treatment is always diet and exercise, and then we start them on metformin. That's the first line medication, and then we move into second line medications. Say with
Even
GLP-1
metformin
agonists.
has a bunch of literature against it, right? I mean, that's not clear.
Sorry, Kristen, I missed that.
Even metformin, there's a body of literature saying it's unsafe. That's not even clear.
Well, for type 2 diabetics, there's a reason why that's the first line medication that we prescribe. It's been used for decades, and the data on that is quite clear that for type 2 diabetics, it does seem to reduce cardiovascular disease in those type 2 diabetics.
Right. Much more controversial for healthy people, right?
Yeah.
To take metformin, right? So there's where you see that it can impair the benefits of exercise, right? And metformin has multiple targets as well, but one of the things that metformin does is inhibit gluconeogenesis, right? So that's why you would take it as a type 2 diabetic. But you don't want to inhibit, complex 1 when you're fit and healthy and exercising.
Yeah, my overall point is that in type 2 diabetics, I want to be prescribing medications that I know will be beneficial for them, and that the benefits will vastly outweigh any negative effects. And when it comes to resveratrol, I'm not a preclinical scientist. When I'm hearing that there's a lot of conflicting data on both sides and that the human data is not conclusive, it's not going to be, resveratrol is not going to be a medication that I'm going to recommend to my type 2 diabetics to take. When it sounds like the preclinical world, just in this discussion, we can clearly see that there is that there are different viewpoints. So I don't know where to take the conversation around resveratrol at this point, because it has been studied for such a long time, and billions of dollars, as Charles has mentioned, has been pumped into resveratrol research, and it's still not coming through as a clear, even type 2 diabetic medication.
Well, part of the problem, right, is that so I agree with you. I don't take resveratrol. I mean, I don't take any of these supplements because it is unclear to me. But that doesn't mean I think I'm 100% that it's bad or right. I just don't know enough yet. And the same could be said for these other supplements that are happening right now, right? So I don't take them either. And I don't think it's that much clearer for them. Metformin being one, rapamycin being one, right? Even NAD supplementation. I don't take any of this stuff because I think it's unclear. How do you?
Well, I'd say the data's clear on metformin for type 2 diabetics, and that's why, again, it's the first line medication that I prescribe.
Well, I mean, that then you talk about the resveratrol literature is pretty supportive in type 2 diabetes as well, right? Like it's coming down on the side of supportive.
Well, there's no guideline that suggests using resveratrol in type 2 diabetics.
Well,
So I don't think that we can say that resveratrol is beneficial at this point for type 2 diabetics.
We can't say that because more research needs to be done, but it's also not patentable. So how do you go forward in getting those trials off the ground? So I think it's unclear. I think more research needs to be done. But, it's also a matter of the system that we're in, and a lot of those molecules are not FDA regulated in the US, right? So you can get anything that claims to be resveratrol and take it. Who knows what it is? So, it's complicated I don't think as physicians, which I'm not practicing, right? I didn't do residency, but I'm a physician, right? I wouldn't prescribe anything that I wasn't 100% sure that is going to have an effect. I wouldn't recommend things that aren't approved, right? Besides exercise, diet, and sleep, and low stress and meditation, like you were saying, right? So I don't know if we should be even making recommendations to people like that.
Well, I suppose it's my job, right? When a patient comes to see me in the clinic with type 2 diabetes, I'm going to treat them because they're my patient.
Right, and I wouldn't recommend stuff that's not approved, right?
Yeah, absolutely. And I think that's kind of the point that I wanted to try and get across, that, billions of dollars have been spent on resveratrol research and analogs of resveratrol. And despite that body of work, it's still unclear as to whether there are true benefits for type 2 diabetics.
You could say the same thing for a lot of other drugs. I don't know what the point of that is.
Well, not for the drugs that are approved for type 2 diabetes that we commonly prescribe.
A little unclear. Even statins are a little unclear. I don't know. I mean, yes, they're approved.
I'm not sure, but metformin for type 2 diabetes, there's a reason why that's the first line medication that we prescribe. It's been well studied, and the evidence is overwhelmingly supportive to use metformin in type 2 diabetes. And then the second line drugs come in with, the GLP-1 agonists, with the SGLT2 inhibitors. Those are the body of evidence is quite clear on that, and that's why they're part of clinical guidelines. And my concern is that, again, despite, and this is just me speaking as a clinician, despite all of this research that's gone on into resveratrol and resveratrol analogs, there doesn't seem to be a clear consensus from the preclinical world, let alone into the clinical world.
I don't know. I mean, has there been more money pumped into resveratrol than other drugs that are approved? I don't know that that's a true statement.
Well, I mean, you could say sirtuin activation had, multi-billion-dollar.
But we already said that sirtuin activation and resveratrol are not the same thing, right?
Okay. Well, that's breaking some ground for YouTube viewers today.
Well, not
Um,
necessarily the same thing, right?
so I I want to maybe pivot a little bit and talk about NAD. So NAD coenzymes, four of them, are the central catalysts of metabolism, and our research has shown that the NAD system comes under attack in many different conditions of metabolic stress, probably 10 different conditions: DNA damage, reactive oxygen species generation in animal models of obesity, type 2 diabetes, diabetic neuropathy, chemotherapeutic neuropathy, central brain injury, inflammation, infection. And through our research into the NR pathway, we found that the NR pathway gets upregulated in a number of these conditions of metabolic stress. So, like at heart failure, cardiac uh, NAD goes down in a mouse. Looks like the same thing happens in human heart failure, and we can show that in a mouse model of heart failure, that oral NR will restore uh, NAD to the failing heart. Nicotinamide won't, because the NR pathway gets upregulated, the nicotinamide pathway gets downregulated in the failing heart. So we think that there's a use case for NR in wellness, as well as potentially in disease and conditions of metabolic stress in which NAD comes under attack. And NR has a great safety profile. There are placebo controlled trials that consistently show safety of NR up to 2 grams a day. And there are increasingly some positive results showing anti-inflammatory effects of oral NR, combination trials where time to recovery from COVID was accelerated, cerebral blood flow in a small Parkinson's trial, and so forth. So I'm pretty excited about that. And but again, I don't get out ahead of my skis and claim that people are gonna live to 150 with it. But I think that the use case is building repair and resiliency as we age, because all of these attacks on our NAD system occur over time.
Yeah, I think overall I'd agree with that. I'm quite excited about the promise or hope of NAD precursors. In particular, I'm excited about their effects with diseased states. So, in particular, heart failure. I think that would be a fantastic trial to run if you could do a randomized placebo-controlled study in class 3 or class 4 heart failure patients. I think that would be a very interesting study. I must admit I'm less certain or less bullish about the effects that it might have for otherwise healthy people. But for disease states, I'm very curious to see the research. Christin, what do you think about the NAD precursor research?
So, I mean, I think that in general, I mean, it's just like everything else. It needs to go through trials, and we'll see what the results are, right? So, I think anything sort of promoted uh, off label, we should be very careful about hype. And I think people should be careful and know that there's controversy in the literature, and to read papers for and against, and to read reviews, and to read meta-analyses, and to make decisions with eyes wide open, and not think there's just one version of any story, which is why I think it's important to have these kinds of conversations, you know. And yeah, I mean, I look forward to seeing what happens with these drugs, and to see where we get. I do think it's going to be very exciting. I think a lot of, there's been the reason there's so much controversy and in all of this is because it's so exciting, for the future. I do think we're going to have drugs that work. I think that we're going to, extend Healthspan at least
Hmm.
a lot, and that people, when they do die, are going to die healthier and, with better quality of life at the end of life. And I'm very excited for that. I'm not going to, I'm going to wait and see for myself and for what I tell other people, in terms of, I'm not going to make a claim that something is definitely going to work or not work. And I think we should all go in eyes wide open to what we're taking and what we put in our bodies, and make sure that we do our own diligence in investigating that, and be careful about what we say, right, in the
Agree.
public. So
Yeah, agreed. Charles, if I may, there's one particular question I wanted to ask you around NAD. There was a recent paper that came out. It was an observational trial that took muscle biopsies from older adults who exercised and compared it to those that don't exercise, and took muscle biopsies from younger people. And what they could see is that there did seem to be a correlation with age and lower NAD, particularly if the older adults weren't exercising. So
Yeah.
what so if in and this is one of the things that I've been trying to grapple with. If people are exercising and leading an otherwise healthy life in terms of diet and meditation and things, that's where I'm a little bit confused as to where NAD precursor supplementation may further improve the
Yeah.
health of those otherwise healthy people.
Well, we all are exposed to some sunlight, right? So sunlight damages DNA. When DNA is damaged, PARP1 and PARP2 get activated and consume NAD in order to facilitate DNA repair. When we all live in oxygen at the intermitochondrial membrane, there's reactive oxygen species that are generated that churns NADPH. So the NAD system is challenged constantly through our life. And when we see double-stranded RNA, or like a coronavirus or Zika, other viral pathogens, we have an interferon system. The interferon system turns on 5 members of the PARP superfamily that consume NAD, and that at that lower NAD state, our defenses against coronaviruses are impaired. That's something that I'm extrapolating that from model studies from our laboratory. So I would say that the NAD system is sort of constantly under attack, as well as is under challenge as we age, and that's the use case for NR.
Yeah, I hear that, and that's one of the big reasons why I'm excited about using these precursors in diseased states. But from that, and again, I'm extrapolating from this human observational study. If people are exercising and having a healthy diet, it seems that they can maintain good levels of NAD in the muscle at least as they get older. So in that instance, what would be the use case to take NR or any other of the NAD
Well,
precursors?
metabolism is trying to do a lot of things at the same time, right? And so if you're repairing, if cells are repairing their DNA, their NAD is for sure compromised. When cells are dealing with uh, activation of the innate immune system through interferon, their local NAD system is compromised. So the athletes, 20-year-old athletes that take NR, are doing it because they can see a performance increase, time to recovery from hard workouts, and seeing benefits. Almost everybody observationally sees anabolic effects, like fingernails and hair growing faster. I shave my head these days, but you see more on the razor, or your hairdresser will see more hair between haircuts. That's observed by almost everybody. And
how do you quantify that, though? I mean, that seems
that's
kind
not, yeah. So that's not, like I qualify that by saying that we don't have placebo-controlled data on that, but that's literally every person that you know, eh, virtually every person notices that, because it's so clear when you're cutting your fingernails
so. Is
and
that
you're
not
taking in.
right?
No, that's not
How's
that's
that
not hype.
not?
That's not
Okay.
hype. That's an observation. No.
How?
Hype would have been if I said that, it'll, regrow baldness, or that you'll live longer, or that your mile time would be faster.
Well,
And I
people,
don't
I
say
mean, a
that.
lot of people want more hair growth, right? So I mean, it's an
That'd be great if it
anecdotal
did, but
piece of
I'm
evidence that's just based on hearsay. I mean, I don't know. I'm just trying to hold you to what you're saying earlier.
Oh, I'm very comfortable with people holding me to what I say. But yeah, the rate of hair growth it's not going to grow hair where you like um I have male pattern baldness like a lot of 60-year-old guys. But it doesn't grow hair where you don't have hair, but the rate of hair and fingernail growth is seen by virtually everybody.
It'll be interesting to see if that stacks up with the placebo-controlled trial, as you say.
Yeah.
Yeah. Christin, is there any other topics that you wanted to cover during this live stream?
No, I mean, I think we got to most of it. I mean, I hope that people can see it, maybe feel good about seeing a more well-rounded picture of why there's so much controversy in this, right? Of how complicated the back and forth of the literature is, and how to be careful and what they take, and looking into it. I recommend going to PubMed. This is how scientists figure things out, right? Go to PubMed, and don't listen to other people, but just type in review article Sirtuin, or review article NAD, review article rapamycin, whatever it is, and just and read the high level literature, or ask someone else about it. I think we should be wise consumers of the things that we put in our bodies, and know the full story. I think it's great to have us on here, and try to give the full picture. But even here, you see that we're not fully in consensus on all of this stuff, and I think that's because that's maybe appropriate for where we are with these stories at this point. So that's all. And I wanna thank you guys a lot for having me on here. It's been really fun.
Yeah, no, I want to thank you, Christin, for coming on. It's great to have someone to offer the other side of the story, and to have an open and honest conversation about it. So I really appreciate you coming on.
Yeah, and to be clear, I mean, I don't consider myself to be the other side. I consider myself to be sort of, a third party neutral observer of what's happening. But yes, yeah, I appreciate it.
Charles, is there anything that you wanted to leave us with, or any other papers you wanted to go through?
Yeah, well, I tend to think that, expert analysis is valuable. I try to be available to people on Twitter at Charles M. Brenner to try to explain experiments. In my view, going through figure by figure is important, and not getting ahead of our skis is really important. And I think metabolism is really exciting, and I want to convey the excitement of that. Um, and I'm grateful to you, Brad, for bringing people together and trying to promote science communication. Thanks to Kristen as well. It's really nice to meet you, and I hope that we can continue to educate people in the future.
Yeah, for sure. I'm super excited about that too. Oh, you can find me on Twitter as well at Dr. Glorioso.
Awesome. Well, I think we'll leave it there. But once again, thank you to both of you. And chat, I really hope you found this conversation useful. Overall, again, can't stress this enough. Diet, exercise, sleep always come first. And always have a healthy skepticism about anything that you may hear from anyone online, including myself. So just wanted to leave everyone with that. Okay. All the
Thanks
very
all.
best.
Yep.
Okay.
Bye.
Thanks a lot. Bye.